Hi everyone, my name is Hussain and the topic of my research is plant uptake of contaminants of emerging concerns in agroecosystem irrigated with reclaimed water. I'm a PhD student working under the supervision of Dr. Daniel Garrity, and the committee members include Dr. Sajjad Ahmed, Dr. Harun Steven, Dr. Dale David, and Dr. Rika Marty. This is the outline of my presentation: problem statement, objectives, methodology, results, conclusion, broader impacts, and references. Starting with the problem statement, basically, water scarcity is a global challenge, and with the increasing population, the demand for water is increasing every day. And about 90% of the available water is used for agriculture. And reclaimed water does give us a replacement for the fresh water, but the problem with the reclaimed water is that it contains a lot of contaminants of emerging concern, including pharmaceuticals, trace organic compounds, a lot of industrial compounds as well. And there is a possibility, if we use the reclaimed water for agriculture purpose, that it could possibly, the contaminants which are present in the water could possibly be uptaken by plants through roots, and then the uptake could depend upon different conditions like growth conditions, soil properties, plant biology, compound properties, and exposure medium. So if the plants uptake those contaminants, that could potentially pose a human health risk. And that's what this research is about, that I will be irrigating my plants with reclaimed water and determine whether there is the uptake of contaminants in the plants or not. So basically my PhD have like 4 different objectives, and for this presentation I'm only addressing one question for my first objective, which is to compare and contrast the differences in uptake and accumulation of contaminants of emerging concerns in tomato and spinach plant structures. So the question I will address here is, does the accumulation vary in different structures of plants? And I'm considering only tomatoes. Methodology includes selection of target contaminants, preparation of agroecosystem, monitoring of plant growth, sample extraction, microwave digestion, and then liquid chromatography, tandem mass spectrometry. Target contaminants. So there are hundreds of contaminants which are present in reclaimed water, but these 13 that I've chosen are actually, the selection criteria is shown in the table. There are different reasons. Some of the contaminants are really resistant to biodegradation. Some of them are present everywhere, and some of them are antibiotic resistant and then cause antibiotic resistance, and then some of them are difficult to remove and then difficult to decay. So there's various reasons for me to choose these 13 out of those hundreds of the contaminants. This table also shows you the physicochemical properties and the typical concentrations. These are the typical concentration in treatment plant in Las Vegas. So tomato and spinach plants were grown in 2 parallel hydroponic systems, but I'm only discussing tomatoes here. The tomato plants were germinated in germination tray in the top picture, and the germination rate was about 63%. We used 2 hydroponic systems. One was irrigated with UF and the other was irrigated with ozonated UF. And we had 10 replicates of tomato plants in both the hydroponic systems. So once the plants were transferred to hydroponic system, the nitrogen, phosphorus, pH, UV absorbance, electrical conductivity, ozone to TOC ratio, water consumption, height of the plants, and chlorophyll, all these factors were monitored throughout the growth period. So once the plants reached maturity, their roots, stems, leaves, and fruit structures were extracted, and then they were further processed so that they can be ready for the liquid chromatography and mass spectrometry to identify and quantify the contaminants of emerging concern in the plant structures. So microwave digestion was used to recover the contaminants which have been uptaken by the structure of the plants. In microwave digestion, high pressure and temperature is applied, and the solid matrix of the plant structures um, are disintegrated, and the contaminants present in the plant structures are taken out using a solvent. After microwave digestion, the solution that we obtain from that process is further used in liquid chromatography tandem mass spectrometry. So before using that solution here, it is purified, and then it is injected into liquid chromatography, where the contaminants are separated on the basis of their affinity for one of the phases, and once they are separated, the effluent from the liquid chromatograph is directed to the mass spectrometer, where the contaminants, they are ionized through an ion source, and then through a mass filter they are separated, and then they're basically separated on the basis of their mass to charge ratio, which help us identify and quantify those contaminants. Moving to the results, during the growth period, nitrogen, phosphorus, and many other factors explained earlier, they were monitored during that period. This graph actually shows the percentage of available phosphorus that was consumed bi-weekly. So first 2 weeks, you can see that for ozonated system, the consumed phosphorus was considerably less as compared to the UF system. But as the plants grow bigger, the consumption of the available phosphorus increased somewhere between 60 to 80% of the available phosphorus. As far as the nitrogen is concerned, you can see that for the first 6 weeks, when the plants were growing up, the consumption of nitrogen was not as much, but as the height and the biomass of the plants increased, the consumption of nitrogen increased as well. And this graph shows the consumption of water over time, and you can see the water for ozonated system that increased from 28 liters per week to 36 liters per week from when the plants started growing to the point when they reach the maturity. And for UF system, the consumption increased from 22 liters to 37 liters. And these are the results that we obtain from LCMS. Basically, it shows us that what are the concentration of specific contaminants in each structure of the plant, and these are the results that actually address our question. So you can see that there are 4 contaminants in the root, which are sucralose, triclosan, sulfamethoxazole, and DEET, whereas where there is like 2 are present in stem, and 6 of them are present in leaves, and 7 of the contaminants are present in fruit. Now it's a little bit interesting to see that some of the contaminants which are not present in roots or stem are also present in the upper part of the plants. And someone would say that, how is this possible? Are they generating during this process? But that's basically because these contaminants do not accumulate in the roots or stem and then they translocate to the next structure. Basically, that does not mean that they're not present in root systems at all. Yes, they're present, but they're below the detection limits. But most of those contaminants move to the next structure, or translocate to the next structure, to be exact. This is the overall picture of my PhD research, and once we have the concentration of the contaminants, I will be able to answer all the questions, but this presentation specifically address the question that's highlighted here. Does the accumulation vary in different plant structures? Conclusions are for this presentation is that the amount of nitrogen consumed is higher for UF compared to ozonated, while the amount of phosphorus is higher for ozonated compared to UF. And then ozonated tomatoes, the structure that I showed, has 4 contaminants in roots, 2 in stems, 6 in leaves, and 7 in fruit. And further, these concentrations will be used to do further analysis, which is still underway. Broader impact. So the results obtained from this research could help increase the acceptability of reclaimed water among farmers, and this research can also determine the potential of hydroponics as an alternative technique compared to soil plant system, and then the data obtained that can be used to determine the potential human health risk associated with the consumption of crops which are grown in reclaimed water. And these are the references for my presentation. Thank you. That'll be it.